All right, gonna go to our next session, imaging cytometry in research. And our first speaker in this session is gonna be Claude. My pleasure. To introduce Claude. He has a degree in biomedical science from the University of Melbourne, Australia. He was introduced to flow cytometry in his first job in 2007 as a lab tech and has not looked back. Claude has worked in both academia and industry and joined the BCM Cytometry and Cell Sorting Core in 2022 as a flow cytometry coordinator. He is responsible for providing the technical oversight of the core, instrument troubleshooting, and data analysis. He has collaborated on a variety of spectral and imaging flow cytometry projects. Recently, Claude has begun exploring the utility of machine learning in cell classification for image flow cytometry data. Thank you, Claude.
Well, good afternoon, everyone. I'm glad to be here. FlowTex is always a highlight for me. As some of you may know, I have 1 million kids at home, so I don't get out much. But when everything comes to me, I take advantage of it. So thank you to the community for having me here. And hopefully, with this talk, I will have piqued your interest about the possibilities of incorporating imaging flow cytometry in your research, whether you're in a core facility, which I am, or if you're in a research lab for your projects. Okay, so before I jump into all the projects, I'll talk to you a little bit about our core. So we are a fairly large core. We serve over 200 research labs. And as you can see from the list of instruments, we have a whole bunch of instruments. And so because this talk is about imaging flow cytometry, I put those first. We have the image stream and the attuned CyPix. We have the S8, the A8, the Aurora, and a whole bunch of other instruments, which I won't cover today. We offer free consultations to our users or to whoever is interested. So we're happy to discuss experimental design, panel design, troubleshooting. We're happy to assist with instrument operation, training, and we're happy to help analyze your data and more. So feel free to reach out to us with any questions, referrals. We're always happy to consult and collaborate within TMC and beyond. All right, so how do we even solicit potential projects for imaging flow cytometry? Imaging flow cytometry is kind of like the ugly duckling in your core. It's the instrument that kind of sits in the corner, gathers dust, gets turned on every now and then for QC, but rarely actually gets used. So how do we get interest in exploring this technology? So we hold educational seminars, user group meetings, webinars. I've worked with a lot of you in this room to organize those, to kind of bring awareness to the technology in our institution and in TMC. We are also chatting with our users during assisted appointments. We ask them questions like, have you ever wondered what the distribution of your protein looks like on your cells? Do you need to know if your protein is in the cytoplasm or the nucleus? Are you sure you're actually getting internalization? Are you interested in spot counting and looking at DNA damage? We also have weekly office hours where we can talk about the concept or the idea of imaging flow cytometry, if it seems applicable to someone's project. And every now and then we get a really engaged investigator that already has read about IFC or has some prior experience with it and wants to try it for their project. So in terms of approaching imaging flow cytometry projects, our general philosophy is to treat them as unique projects because they are unique projects. They're not your run-of-your-mill sorting appointments or analysis appointments. The instrumentation is different and acquisition software is different. Data analysis software is different. The data itself is different. So there's a bit of a learning curve when users want to embark on the journey of imaging flow cytometry. This appeals to some people, others not so much. However, it's important to communicate that there's a broad range of discovery that can happen with some relatively simple experiments. This is useful as a way to overcome that initial hesitancy to IFC. And as you can see in the projects I'll describe, the majority of the experimental setup is actually fairly straightforward. And even with that, we're able to extract a lot of information from fairly simple experiments. Generally, we have a lot of consultations with users thinking about imaging flow cytometry. These can happen before you start your project, during the project, when the wet lab experiments are happening, and post-acquisition and during data analysis. We offer a lot of support for data analysis in particular, because that is probably the part that's the most different to standard flow cytometry, where users can have fully assisted analysis, basically done by us, partially assisted, or we can kind of throw them to the wolves and, help them along the way. We have a lot of consultations with them. We have consultations. Did I say we offer consultations? So in other words, we will walk you through every step of the way as much as possible, because this is a technique that requires that. On a side note, we now have some users that are starting to take advantage of large language model AIs like Copilot, ChatGPT, and that's actually dramatically improved the capability of custom data analysis outside of vendor-provided software. So that's something to keep in mind, something that wasn't even available really a year ago or widely even thought about, that if you're approaching IFC and you have a question that maybe you can't tackle with the vendor-provided software, you can start delving into these large language model AIs. Okay, so I'm going to be focusing on two instruments today, the Cytek Image Stream Mark II and the Attune Cypix, and some projects that highlight their use in the core. So let's start with the image stream. So this instrument has been in the core since 2016, well before I joined the facility. 4 lasers, has up to 60x magnification, up to 10 fluorescent channels, and as Rob covered this morning, you can use it for a whole bunch of different applications, which I won't go into. In my mind, this is still regarded as the gold standard imaging flow cytometer based on image quality, number of fluorescence channels, and the relatively easy to use data analysis software that allows you to do very powerful analyses on your data. So the first project I'm going to highlight actually reaches back to my previous institution at University of Virginia, where I worked with Steve Zeichner's lab, and we used the Cytek image stream as a proof of concept for the production of E. coli-derived mini-cells. So what on earth are mini-cells? They're produced by bacteria with mutated MIN genes, and this mutation of the MIN genes results in cell division instead of happening in the middle of the cell and kind of resulting in 2 equal bacteria that come from that. It forces the septum to develop towards one of the ends or one of the poles. So it results in a large cell where that has copies of the bacterial chromosome and a small mini cell that lacks chromosome but still is metabolically active and still contains some protein or whatever other materials that you could put in there. So we wanted to be able to show proof of concept that these mini cells were a potential transport mechanism for drug or vaccine delivery. The long-term goal would be to have genome-reduced E. coli that could be dedicated to the production of a particular drug or vaccine. The advantages of the system are that all the work is done by the E. coli. They're relatively easy to handle and easy to grow. All the material is concentrated and packaged into the mini cells for delivery, and it's fairly cost-effective, especially for developing nations. So after the Zeichner lab generated these MIN mutant E. coli, we used IFC to demonstrate the presence of these polar and apolar phenotypes. So these E. coli were engineered to either produce GFP or influenza hemagglutinin, kind of as proof of concept. And basically, we were able to show that we do get polarization. Obviously, at this stage, it's still a mixed population because they haven't really been refined. But the polarization indicates that these bacteria have the potential to produce mini cells, where the division starts forming towards one of the poles of the cell versus in the middle. So this was great. This proved to us that we were able to generate these mini cell producing E. coli, and we could prove that using imaging flow cytometry, or demonstrate that. So then we also used IFC to demonstrate that we could basically identify these mini cells using scatter. So the high scatter events being the kind of elongated, larger bacteria, and then the low scatter particles being the mini cells. Then we also demonstrated that the protein concentration in the bacteria was versus the mini cells was actually higher in the mini cells by performing a signal intensity per area calculation using the IDEA software. And we showed that in the non-genome reduced E. coli versus genome reduced E. coli, that the signal intensity is higher both in the mini cells, which are these peaks here on the right, and also the genome reduced peaks here, were higher than the non-genome reduced. So after showing that we could identify mini-cells from larger bacteria and that we had a strain of E. coli that could produce the mini-cells and produce the protein that we wanted, we used IFC to verify that there was actually the protein of interest in the mini-cells. And so you can see in both the genome-reduced bacteria and non-genome-reduced bacteria, you get this green, beautiful green signal, GFP, or labeled hemagglutinin. And so this served as an important proof of concept for the potential use of these mini-cells as a vaccine or drug delivery mechanism. Next, I would like to highlight some work done with Dr. Katie Patras. So she actually presented this here at FlowTex in 2024. I still wanted to highlight this as an example of using IFC in research. And if you'd like to learn more about her specifics of the UTI model and the biology, you can go back and look at the FlowTex YouTube page and her talk. So obviously, I'll be focusing on more of the technical aspects of imaging flow cytometry. And as a brief overview, she had found that the presence of TAM horsefly protein, or THP, was critical for allowing neutrophils to undergo netosis. So netosis is where neutrophils form nets, or neutrophil extracellular traps, and they do this by releasing DNA into the extracellular space to trap and immobilize pathogens. So from mouse studies, she had determined that the THP was critical for neutrophils being able to undergo netosis, and wanted to use IFC to image human neutrophils to show the same effect, but in human samples, or at least that's how the project started. So after a few pilot experiments on the image stream, Dr. Patras, being curious and taking the initiative upon herself, she went and started digging in the literature to see if anyone else had looked at netosis by IFC. She stumbled across this paper by Leliot, where they had actually already used the image stream to characterize netosis into 6 phenotypes that you can see here. So after we discovered this, we thought, okay, let's try and reproduce this analysis in the context of urinary tract infection. So after some more experiments and more optimization, we basically report that, yes, we were able to successfully reproduce the analysis from Leliot et al., which was amazing because, as David mentioned, reproducibility is kind of a problem in science. So the fact that we could kind of plug and play their analysis into our system was amazing. So we were able to reproduce the 6 phenotypes that you can see here in their figure and in our figure here, and basically able to demonstrate that netosis seems to follow a fairly defined set of phenotypes across 2 different systems. So we'll revisit this later in the talk. So onto our second instrument. The Attuned Cypix was installed in the core in February 2024 as part of a long-term collaboration with Thermo. It has 14 fluorescent detectors, 20x bright field imaging, and has acoustic focusing, which allows you to run samples really, really, really fast and still get good resolution of your data. However, I don't think you should run them at the maximum flow rate. But it's there. Initially, we needed to determine the quality, the imaging quality of the instrument, given our long-standing experience with the image stream, and our goal also was to just put it through its paces in a core. So our early uses of the instrument were basically to compare some data sets side by side with the image stream, use it for some basic sample prep QC and pre and post-op QC to basically show that, oh, you are actually isolating cells, viable cells, and not just garbage, and you actually sorted cells and not just garbage. So that was kind of our first use cases. Since then, we've kind of developed our uses a little bit. We've got some specific imaging projects, which I'll cover, and it's also routinely used as an alternative to NXT. Fortunately or unfortunately, that happens to be the majority of usage for this instrument, but hopefully we'll get some more interest as time goes on for specific imaging projects. So here's some advantages, disadvantages of the Cypix. And I want to highlight this, again, kind of using the image stream as a comparison. So the advantages are that it has high throughput at your standard flow cytometry rates. You're running at the same rate that you'd run on any other cytometer. has acoustic focusing. You can use it as a non-imaging cytometer if you really want. It has really high image quality, which you'll see in a few slides. And it's very easy to use, especially if you're familiar with NXT, and there's no special sample prep required to use the instrument. You don't have to change your concentration, you don't have to change your tubes, you just basically prep them like you would any flow assay, put it on and run. So disadvantages are that there's no fluorescence with the imaging, and the imaging resolution is limited to 20x, and there's no spectral capabilities yet, although I hope that that is in the pipeline for Thermo. So here's some representative images comparing the image quality of the Cypix against the image stream. So because the image stream, as I said, gold standard imaging flow cytometer, we wanted to validate this against that. And so you can see in a variety of cases, like this is a cancer cell line, you've got your Cypix image on the left, image stream on the right. They're very, very comparable. Cypix does very, very well, especially considering the flow rates that we're running at. Here's an example of E. coli on the Cypix and E. coli on the image stream. Again, very comparable. Some yeast and a PBMC. So you can see bright field imaging quality on the Cypix is pretty darn impressive, in my opinion, considering how fast you're running. So as a proof of concept for validating the instrument, we decided to use just a standard flow cytometry assay, an Nexon 5 PI apoptosis, to demonstrate the ability of the instrument to distinguish different stages of apoptosis using conventional flow and see if we can get any kind of enhancement or benefit from having imaging. So as you can see from this figure here, there are a lot of morphological features that happen to a cell when it undergoes apoptosis or necrosis that could be distinguished by imaging. And Radhika Pradhan from the Justin Lee Lab volunteered her data for this validation. And so thank you to her. So we basically took these neuroblastoma cells and she was titrating them for kind of a positive control in this particular assay, treating them with hydrogen peroxide or cobalt chloride, and then looking at different concentrations, different time points for Nexon and PI. So you can see on the bottom, our gating strategy is pretty standard. We've got scatter, we've got our doublet discrimination gates. And then where it starts to differ is over here where you have some parameters that are specific for the image processing. Basically, can the software identify these cells as cells? And based on that imaging, what is a single cell? What is more than one cell? Okay. And so for the fun of it, we decided to see if we could analyze the data the traditional way, which you can kind of see here in the middle, as well as use some of these new clustering and dimension reduction features in the Cypix software and kind of do a parallel analysis. So the Cypix software allows you to do some clustering, as you can see here. Basically just divides it into either user-defined clusters or automatic clusters, and also lets you create a UMAP. So you can see that based on the high dimensional analysis, it divides up really, really distinctly, and these groups of cells and these clusters of cells overlay really nicely with the traditional analysis. So far, so good. And as you can see, looking at some of the representative images of each of those clusters, the populations line up with your kind of classical bivariate analysis. You've got cluster 4 that lines up with your nice round viable cells, cluster 1 that lines up with your early apoptotic cells, where I don't know if you can see from up there, but you can start to see some nuclear condensation inside the cell. They lined up so well, in fact, that I had to be careful not to select the same representative images from each gate. That's how well they lined up. Cluster 2 corresponds with late apoptosis, and then cluster 3, necrotic. I'm sure someone out there who knows more about apoptosis than I will argue with me about all these, but basically this is just a proof of concept to show that the Cypix is capable of both traditional flow cytometry analysis and using some of these high dimensional tools and taking advantage of imaging. It's a great way to confirm your populations of interest. So on to another project. We have a user in the core, Benjamin Belfort, an MD PhD candidate. Well, He's done his purgatory in the lab and he's back in the clinic. So he was looking for a way to QC his sample prep for sorting nuclei in our facility. And he kept having problems, couldn't quite get lots of really viable nuclei. So the reason was he decided to use the tissue from the mouse olfactory epithelium, which is kind of a difficult tissue to work with because you've got all these bony structures in there that kind of divide the air up, humidify it, that get in the way. So he worked with Miltenyi and developed a GentleMax protocol to isolate these reasonably well. And then we threw them on the Cypix and basically QC'd his sample prep. So what you see here is actually one of the newer features of the software too, where it allows you to train a model to basically segment or mask your cells of interest. And so when we did that, we were able to come up with a model specifically for his experiment with about 98, 99% validation accuracy and training accuracy. So you're probably wondering, okay, what is UDT model training? We were asked to participate in the beta testing for that. And basically, it allows you to define what's a cell and what's not. So with the early versions of the Cypix software, you're kind of stuck with whatever annotation models they had built in. And on the left, you can see they do okay for round, regular cells. But even then, if there's something weird with the background, it'll start annotating those as an object. And then when you have something like a sperm that's irregularly shaped, it doesn't do well at all. In fact, it misses the whole tail. So after refining those UDT models, you can see on the right, the annotation is much more accurate to the objects being acquired. So back to Ben's data. Here's the gating strategy we used to identify his nuclei. Again, very standard, scatter, singlets, DAPI positive nuclei. And then we started to use some of the imaging parameters to QC his nuclei. We found two parameters that were useful. Circularity, which as the name suggests, measures how round or circular an object is. The closer the value to 100, the more circular. And eccentricity, which measures how oblong an object is. And the closer the value is to 100, the more oblong an object is. So here are some representative images from high circularity events. You can see the nice round happy nuclei, and then high eccentricity events where they're kind of oblong and unhappy and falling apart and no good. So these parameters allowed us to get a very clear distinction between viable and non-viable nuclei. And happy to report that this was actually published as part of a Jove article or Jove video as of August 2025. I encourage you to check it out. The videos of the dissections are very cool, very interesting, and the flow plots are not that interesting, but very, very happy about that. Next, we have an ongoing collaboration with Dr. Blair Stocks, and he approached us with an idea for a sperm classification project. So the classical method of kind of determining viability of sperm is manual. It requires an expert urologist to kind of look at a smear of just a few hundred sperm on a slide. We wanted to be able to automate the classification by using large numbers of images, with the idea that having lots of images would then lead to more accuracy in classifying sperm as viable or not. So again, we used the Cypix to develop a user-driven training model and then manually gated to explore what populations of sperm there were. So after that, we actually were able to identify 8 different populations using some manual gating, and 2 parameters stood out as being useful to us. The skewness intensity on the y-axis, which refers to the distribution of light and dark pixel intensity in an object, and the radius of gyration on the x-axis, which refers to the compactness of the image. So how these kind of separated out was populations 1 through 4 on the top, which are higher skewness, tended to have round heads, and the populations 5 through 8 tended to have small, more conical heads. And as you increased in gyration radius, that the sperm actually got more elongated. We also noticed that there were differences between fertile and infertile samples, that there were less of populations 5 through 8 in the infertile samples. And this lines up with clinical observations where talked about the head shape. Sperm that tend to have that more conical shape tend to correlate with better fertility. And here are some representative images showing those differences. So populations 1 through 4, high skewness. Actually, well, I guess you can see it from up there. The head shape is definitely kind of more on the rounded side. Population 5 through 8 have the more conical heads. And then as the gyration radius increased, you can see that these tails go from being really curled up to being nice and straight and elongated. So that was a nice way to classify those sperm. Obviously, further work needs to be done. We need to, figure out, okay, what are these A populations? Are they meaningful? Kind of along the lines of this figure here, which shows all the kind of morphological abnormalities that can happen. And obviously, to actually automate the process. We've automated some of the process in just being able to accurately identify sperm, but then we need to be able to, put them to different buckets. Is it phenotype A, phenotype B, phenotype C, et cetera, et cetera. So we have a lot of data to work with. We have about 2.8 million images per plate, and we ran multiple 96-well plates. So it's just having the time to do it. And we're hoping that with more images to classify, that we will be more accurate in our classification versus the classical method. Okay, so let's revisit Dr. Patra's work. So as I mentioned previously, we were able to manually reproduce this previously published analysis by Leliot et al. Using fluorescence labels and analysis methods outlined in their paper, as you can see here, which also was published as well. This raised the question, could we do this label-free? Could we do this on the Cypix without, relying on the fluorescent staining? So what we did, again, we trained a user-driven training model, and we used about 730 images, and we're still kind of in progress. You can see that there's been a lot of trial and error, right? Because we have a few of these morphological phenotypes that are very similar to each other. Types 1, types 2, 5, and 6 are all kind of round, and they differ really by nuclear morphology, which is kind of difficult to identify by Brightfield. It's still in progress, but we were able to get a decent accuracy. We're in the kind of 80-ish% training and validation accuracy. So there's still a little bit more work to be done, but I think we're at a pretty good place so far. And so after all that trial and error, we determined that, yes, we are able to use just Brightfield imaging parameters to distinguish these 6 phenotypes of netosis. So we found 2 parameters again, entropy co-occurrence intensity and gyration radius, like with the sperm. And these proved useful in distinguishing the morphological phenotypes without the use of fluorescence. So the entropy refers to how disordered the pixels are within an object. Theoretically, the cells that have more defined features are supposed to have lower values, and the cells that have less defined features have higher values, but we actually found the complete opposite because our healthy cells tended to be highest entropy, and the cells that are kind of apoptosing and undergoing netosis tended to have the lowest values. And as with the sperm, radius of gyration refers to compactness, and so that was particularly useful for distinguishing the nets and the DNA fragments, which are no longer round cells, but kind of spread out, diffuse structures. And then here are some representative images comparing what we determined with our published analysis on the image stream using fluorescence versus the Brightfield features on the Cypix. So you can see they actually line up pretty nicely. We can get our healthy neutrophils here, the type 1, type 2, where their nuclei are starting to decondense, type 3, where they're basically undergoing netosis and spewing out DNA. Type 4 is just the DNA itself, kind of free-floating. And then type 5, condensed nucleus. Type 6, kind of diffuse, also not happy cells. So the question is then, great, we can now reproduce a fluorescence analysis using Brightfield only. We're quite happy with that. Now, are we able to get some kind of automation to this classification? So then we used Amnis AI. So for non-coders like myself, Amnis AI actually offers a user-friendly interface to kind of harness the power of CNN and random forest machine learning algorithms to classify these data sets. Don't ask me how those work. I'm not a machine learning expert or a coder, but it allows people like me and maybe people like you to use this in a user-friendly manner. So we set out to train a model using the Amnis AI for these neutrophil phenotypes. As with the Cypix model training, there's quite a bit of trial and error, basically an iterative process of, initial training, going back, reassigning cells that get misclassified, retraining, doing that again and again and again, and until you arrive at a model that's kind of, relatively satisfactory. We attempted to train models, first using the fluorescent staining, kind of as we would our original data, and then second, using the Brightfield imaging only parameters from the image stream, since we had demonstrated we could do that previously. So we were able to train a model using the fluorescent staining, which you can see here, matrix A, which shows kind of the probability of being able to classify each of these 6 phenotypes accurately. And then we tested that on an unseen data set, which is here, matrix C. So we arrived at about 80-ish% probability of accurately classifying cells, which we're quite happy about. Then we used the Brightfield data, which you can see in matrix B and D, B being the training set, and then D being the classification of an unknown data set. It had a little bit of a harder time classifying the unknown data set, just like I mentioned, because you have some morphologies that are phenotypically similar, and if you don't have the DNA staining, the software has a hard time deciding which cells go where. So we're kind of in the 70-ish, 80-ish% probability range, which is not bad. Still could use some work, but I think it's definitely a possibility to use Amnis AI to classify Brightfield only. And with that, I'm done. So I've hoped that I've shown you through a survey of different projects on the image stream and the Cypix, the value of using imaging flow cytometry when it comes to a core setting or in your research. And I'd like to thank a bunch of people, obviously the core that I'm in, and Joel, a director, the Zeichner lab at UVA, and the Lee lab, Renkyo lab, the Stocks lab, the Patras lab at BCM, Brian Hall from Cytech Biosciences, who patiently kind of walked me through the Amnis AI workflow again and again and again, and the wonderful people from Thermo that helped me along with the Cypix analysis. So with that, I'll take any questions. You either understood everything I said or nothing at all.
Well, I'll say, first of all, you were very clear. So you're getting these sort of like morphological categorizations for stuff like sperm. How do you anticipate that could be taken then into the clinic, right? Because clinics don't have imaging cytometers, but how could that interface to bring to patients?
Yeah, so I mean, that would be kind of the ultimate goal, particularly of Dr. Stock's group. Obviously, we're still in the very early stages, but the goal would be to put something like a Cypix or something high-speed Brightfield imaging in a lab where you can kind of just throw a sperm sample on, run it really quickly, throw it through a model, and have it spit out basically, classification for a clinician, right, to then sign off on or figure out. So yeah, that's kind of the pie in the sky goal, but we're at the very, very early stages of that.
Thanks.
Okay.
Hello.
Yeah.
Okay. Great talk. I had a question about acquisition time. So when you're looking at specific cells that are more sensitive, like neutrophils, the longer that they're not in a happy environment, they can start undergoing apoptosis or netosis. Have you had issues with that, with more sensitive cell types or samples, and how long the samples will take to run? And
Yes,
either
so with the netosis samples, there's some processing time involved. So basically, the Patras lab, they would get the blood samples from some place and then process them right away and then bring them to the instrument. We'd run them on the image stream. The image stream acquisition times kind of vary depending on this concentration. Sometimes we were done in an hour with, say 5 to 10 samples. Other times we were literally sitting there for 3-4 hours watching grass grow, as one event per second went through the instrument. It didn't seem to affect the data though, like the trends carried on between like over the experiments, regardless of whether we took a long time to acquire or not. When we ran them on the Cypix, it was like a night and day difference. I was basically done in about 5 minutes running the samples. I had a few, single stain controls that I need to run when we were using fluorescence for that. But then running the samples was roughly about 5-ish minutes a sample, and then I'm done. So.
got it. Thank you.
Any other questions? I won't bite. Okay. Thank you.